In this video, what we're going to do is walk through the beginning of the fundamentals of biochemistry that you will need to recall in order to be successful at chemical biology. So what we're doing in the next several videos is building a really strong foundation so that you're ready to tackle the applications that we'll see throughout the rest of the semester. Beginning with proteins, when we are looking at proteins, we need to first break it down and think about the building blocks of proteins. And the building blocks of proteins are the amino acids. Those are the monomeric units that are going to link together via covalent bonds to form proteins. So in the next few videos, we're going to look at the structures of amino acids, some ways of classifying them that will be important in determining ultimately the function of the protein. We're going to look at mechanisms of how bonds form between amino acids to yield proteins. And then once we formed a protein, we're going to look at the different levels of organization of the structure of proteins to evaluate the function of those proteins. So let's go ahead and get started with looking at the structures of the amino acids, the building blocks of proteins. And in order to do that, what I'm going to do is get the screen share going here so that we can follow along on the whiteboard app. Additionally, there is under modules for module 1, you will find that there is a handout on amino acid structures that would be handy to refer to here. It would also be handy to have when you go to do the quiz and the worksheet related to this unit, because you will likely need that information, and maybe you remember it from your biochemistry course, maybe not, but just in case, you can have that out for the quiz. So we're looking at the building blocks of proteins here. And the general overall structure that we're going to see for each and every amino acid is that every amino acid is going to have kind of the same fundamental structure, which is that it's going to have a carboxy group, which we could fill in as COOH for now. Then we will have an amino group, an amine functional group. And then we have the R group, which is the variable component of each of the 20 standard amino acids. And then we have generally a hydrogen right here. In the case of amino acids at typical pH of 7, what is actually the case due to the pKa's of the functional groups in this general amino acid molecule is that rather than having a carboxylic acid group COH there, the pKa of a carboxylic acid group, being that it's relatively acidic, is around 3.5 or so in general. And so what that means is if the pH is above the pKa value of 3.5, that carboxylic acid group is going to be primarily found in a deprotonated state. So if the pH is, say, 7, that is the typical physiological pH, what's actually going to be present here is a negative formal charge. Rather than having the carboxylic acid group there, it's going to be the carboxylate, the deprotonated carboxylic acid. Similarly, if we look instead at the amino group on the other side, the pKa of that group is around 7.8 or so. And so what's going to happen there, since the pH, typical physiological pH of 7, is less than the pKa, that group is going to tend to be carrying around an extra proton predominantly. So rather than being NH2 there, it's actually at physiological pH going to be predominantly present as NH3 with a positive formal charge there. And so the consequence of that is you end up with amino acids that are going to be found as so-called Zwitter ions, whereas Zwitter ion is a compound that contains both a negative formal charge and a positive formal charge in the same molecule. And so the net charge on the molecule is 0 because you have that positive and negative formal charge, which I'll abbreviate as FC, in the same compound. And the fact that amino acids are found as Zwitter ions at physiological pH is going to have important implications related to the function of these molecules within the cell and things like their water solubility. The majority of proteins are water soluble because of the fact that they have these negatively and positively charged amino groups and carboxy groups. So continuing to look at amino acids beyond thinking about what the formal charges would be and some of those general structural features, one important consideration with the amino acids is that the 20 standard amino acids are all the L configuration. What we mean when we say L stands for left or levorotatory, meaning that they are going to rotate polarized light to the left. So if you were to put them in a polarimeter, they would rotate light to the left. They have that specific chirality, and that chirality is going to be essential later on when we start thinking about the structure and function of proteins, because once we join these L amino acid building blocks together, that very specific chirality is going to mean that the overall protein structure will be specific for recognizing molecules that interact with it that are a particular stereochemistry as well. And so we'll go ahead and add that to our bullet point list here about things that we need to keep in mind related to amino acids. And that is that the chiral amino acids are all the L configuration. And you might be saying, now, wait a minute, I think I've heard some things about D amino acids. That would be the enantiomer of the L amino acid. And you're right, if you're digging into your brain reservoir there and thinking of D amino acids, there are examples of D amino acids, particularly in bacteria. Bacteria are sometimes capable of constructing cell walls that are made of D amino acids rather than L amino acids. But the bacteria have very specialized ways of creating those D amino acids within the cell, and the typical building blocks of most proteins that we see are going to be the L configuration. And that is going to have important implications about how the protein behaves. Of the 20 standard amino acids, I mentioned here that the chiral amino acids are all the L configuration. There is one amino acid, though, that is achiral, that is neither L nor D because it doesn't have any chiral centers. And that amino acid is glycine. Glycine is the simplest amino acid. It is achiral. And we can take a look at the structure of that if we go to our amino acid slide here. And you will see that in our typical amino acid, the chiral carbon is the carbon right here that I'm putting a star on. Remember, chiral means 4 different groups attached to it. Glycine is the only example of an achiral amino acid of the 20 standard amino acids, because if we look at this carbon right here, we have 2 identical groups bonded there, the 2 hydrogen atoms here and here, so therefore the glycine cannot be chiral. So that is the only amino acid that does not have the L configuration of the standard 20 amino acids. So looking further at these amino acids of our 20 standard amino acids that are present in the handout, we have some specific structural features that we can use to categorize the amino acids. And when we start linking these together, a video or 2 down the road from now, we are going to see that the particular link that we have is going to determine the structure of the protein in addition to the nature of the R group. And the R group in this handout is what's highlighted in pink in all of these. That pink highlight represents the R group that we have there. And we can classify the amino acids and predict their functions based on the nature of the R group. So if we take a look at the handout outlines some of the classifications that we can think about. One of those classifications is that we can think of proteins, or we can think of the amino acids rather, as being either aliphatic, and aliphatic we can also synonymously refer to as being hydrophobic. Those are going to be amino acids that, when we link them together into proteins, tend to bury themselves within the interior of the protein so that they are as far away from water as possible, because they have hydrophobic R groups. They have R groups that are comprised primarily of carbons here, looking at all these carbons being relatively non-polar groups, we have carbon-hydrogen bonds. Those groups are not particularly polarized, making these groups as a whole very hydrophobic. On the flip side of things, we also have a set of amino acids that we could classify as polar. And the polar amino acids, we could describe, rather than being hydrophobic, as being hydrophilic, water-liking or water-loving, as the term philic refers to loving. So the polar groups are going to be comprised of bonds that have some heteroatoms mixed in, particularly ones that are going to introduce polarity into the molecule are oxygen atoms, such as you see in serine and threonine, sulfurs, such as you see in cysteine there, particularly sulfurs that are capable of hydrogen bonding because we have that SH group, analogously behaving to the hydroxy groups of threonine and serine. And we also bring in, to some cases, groups such as amides for asparagine and glutamine. And we can bring in say, proline. We add some polarity in there as well. So we classify those as our polar amino acids. We can also break this down into some other categories like aromatic amino acids, due to the fact that they have an aromatic R group present there. And that aromatic R group, the fact that the aromatic ring is planar due to the sp2 carbon atoms that are in that aromatic ring, are going to confer some specific properties to that portion of the protein that has aromatic amino acids. We can also look at amino acids that we would describe as acidic or as basic. And acidic and basic amino acids commonly play roles in catalyzing chemical reactions, because one of the common things that has to occur during a chemical reaction is a proton transfer, a proton being gained or a proton being lost. If you think back to your organic chemistry, that was a very common step of a bunch of different reaction mechanisms that you looked at in that organic chemistry sequence. And so enzymes catalyze reactions by donating protons or removing protons as one of their common ways that they go about doing things. And so we can think of our basic amino acids as being ones that have amine R groups. Remember that bases have a propensity to accept protons. So our proton acceptor R groups are going to be arginine, lysine, and histidine. And we could think of these as being our basic amino acids, because during chemical reactions, they are going to tend to go from having, say, an NH2 group here to an NH3. If they start off with an NH2, in the case of lysine, for example, as I'm drawing out here, and there is a proton to pick up somewhere else. Over here, I'll put it in green. What happens is that the basic nitrogen atom comes in, grabs that proton, forcing that bond to the proton and whatever group it's bonded to there, such as an oxygen, to break. And that will result in that NH3 plus there. So basic amino acids are generally capable of gaining a proton to yield a favorable reaction product and help catalyze reactions as a result of that. On the other hand, negatively charged R groups, also known as our acidic R groups, are aspartate or aspartic acid and glutamate or glutamic acid. And what happens in these is that they have a propensity to lose a proton. So if they start off with this R group that has a carboxylic acid group at the end, what happens is that they come into contact with a base that will remove that proton to create the deprotonated ion there, the negatively charged R group, as a product of that reaction. So these are some of the general features of our 20 amino acids. And what's going to happen is that by joining links between the amino acids to make a protein chain that then folds up into a three-dimensional structure, the individual R groups of these proteins, the individual R groups of the amino acids that make up the proteins, are going to drive the ultimate function of the protein. It's going to drive how the protein is able to catalyze a chemical reaction. And one of the things that is looked at quite a lot in the field of chemical biology is small molecules that are formed through enzyme catalyzed chemical reactions, and how those small molecules that are formed through these enzyme catalyzed reactions function within a cell. And so we need to be able to understand how the enzymes function, and that goes back to the amino acid structures, making this a super important concept that we need to be familiar with. So what we are going to do in the next unit is we're going to be continuing this discussion of amino acids by looking at how we can link those amino acids together to form longer chains. So we will get into that in our next unit.